Welcome again, my name is Evans Matuga, your usual biology teacher. Today we are learning about reproduction. Now, in reproduction as a topic, we'll embark on a subtopic known as mitosis. But before we look at mitosis and understand how the processes occur, it's important to refresh ourselves about a nucleus. We have learned about a nucleus all our times from Form 1 up to Form 3. And this nucleus has a lot of things in it. But the most important thing about the nucleus is that a nucleus controls all the cell activities. So it controls activities of the cell. And that means without the nucleus, there is no meaningful thing that can take a place in our living organisms. If you remove it, then there will be no life in that particular individual or living organism. Now, a nucleus as it is surrounded by a membrane known as a nuclear membrane. Inside that nuclear membrane, there is a nucleolus. So therefore, I want now, apart from that one again, to understand that a nucleus as it is, how does it control the cell activities is what is important in this lesson. And therefore, a nucleus has got thread-like structures known as chromosomes. And therefore, these chromosomes cannot be seen using even a microscope at that point. So how does these chromosomes look like is what is important in this lesson. And if you look at this point here, this is a chromosome inside a nucleus, though it is not visible, but after some time, after the organization has taken place, these chromosomes is seen as having two parallel chromatids. One is called a chromatid, another one is a chromatid, and at the center here they are conjoined by a centromere. So these chromatids are very vital in controlling all the activities. At some points here, there are locations called gene loci, in singular gene locus. So at these points here, there are also genes which are carrying DNA material. So they carry a deoxyribonucleic acid. It is that deoxyribonucleic acid that actually has a coded information that controls the characteristics of an individual. So an individual will have a particular gene, a gene for tallness, a gene for brightness, and very many things. Keratin, that is responsible for the growth of your hair, and very many other things. So anything that you see in yourself as an individual is being controlled by a particular gene that has a DNA that is attached to that characteristic. So that is how important a chromosome is that is inside this nucleus that we are talking about. So in the entire time we are going to speak about the mitosis process, we must mention the chromosome at some point. So it is very important therefore to understand this chromosome we are talking about. Where is it, and how does it function is very important. Now, after understanding that one, it's now also very important to understand that the cell division, which involves mitosis and meiosis, takes place in specific locations or points. Cell division does not just take place anywhere. So the cell division we are talking about takes place in isolation, in some specific points. Like if you look at animals, cell division will take in Malpichan layers of your skin. There is a layer of your skin. There are many layers on your skin, including the confide layer, the granular, the Malpichan. So specifically, it takes point at a layer called Malpichan layer. In other living organisms, in an ovary, it is in the germinal part. In the testes, there is a specific part called seminiferous tubules. So that is where cell division will take place. If you go to plants, because biology involves animals and plants, in plants there are also specific locations where cell division will occur. So cells actively divide at some points called meristematic apical regions. These are apical meristems that involves the shoots and the roots. And so therefore, when we are speaking about the shoot and the root, they are not enough in answering a KCS equation. You must tell us that the apical meristems involves shoot tip and line tip, and root tip. So it is shoot tip and root tip, not the whole root, not the whole shoot. So that is to say plants grow specifically on their root tips and their shoot tips. If you look at some other points like the bases of the leaves, the internodes, they also are involved in this cell division. So that is where these processes called mitosis and meiosis are supposed to take place. And now that we have said, today we are concentrating ourselves on mitosis, it is important now to understand what the process is all about. Now, embarking on mitosis, it involves several phases which start from a particular phase to the last phase. And the first phase, which is the first stage, is known as interphase. The next phase is known as prophase. The third one is known as metaphase. The fourth is known as anaphase. And the fifth one, which is the last, is telophase. So now, at this point in time, it's very important in this lesson to understand these phases we are talking about, what describes them. How do they look like? How will I be able to differentiate between interphase and prophase? In an examination, that is what most important in this lesson. So we'll be able to learn through them, but as we also learn, be able to distinguish between an interphase and a prophase, a metaphase, etc., etc. So let's start with the first one, which is interphase. Now, the first one, which is interphase, has a lot of activities which happens there before the processes start. So that is why, therefore, we call interphase as a resting phase. It has these following activities. Activity number one, there is the replication of the genetic material, that is the DNA, which replicates. And number two, there is the synthesis of new cell organelles. Number three, there is build-up of energy in form of adenosine triphosphate. I repeat, number one is the replication of the genetic material, which is the DNA, and number two is the synthesis of new cell organelles that involves, number one, the mitochondrion, number two, the Golgi apparatus, number three is the centrioles. So you shall see why those formation of organelles are very important as I take you through the stages. And finally is the buildup of energy in form of adenosine triphosphate. We usually write it as ATP, but we discourage you from using ATP and encourage you to use adenosine triphosphate when answering a question. So now, let's go back to our phases and start from the first one and see how it appears. Like at this point, an interphase, and at this point, which is the interphase, you are able to see some structures. And as I told you, this is a cell. And a cell at the outermost is a cell membrane. At this point here is a cytoplasm. And then, as usual, this is inside a nucleus. And this nucleus is covered by a membrane known as a nuclear membrane. And inside the nucleus, there is a nucleolus. And inside the nucleus here, there are structures that you cannot be able to tell what they are, but those are chromosomes that are not visible at this stage. And after understanding that one, I've already mentioned that at this point there is synthesis of new cell organelles, which involves the mitochondrion. Of course, we need energy in the whole process from interphase to telophase. Number two, we need the formation of spindle fibers because centrioles are very vital in the formation of spindle fibers. I'll tell you why we need the spindle fibers at these stages. So whenever you see a diagram like this, this is a stage called as interphase. How you can identify it is the chromosomes are not visible, and you can also be able to see the nucleus and the rest. Now, after understanding this one, after the process has already started, we now proceed to the next one, which is the prophase. At this point, which is the prophase, you are able to see some things. One, there is the nuclear membrane, which is still there. There is the nucleolus, but at the center here now you can be able to see these are two parallel strands, chromatid. This is now a chromosome that we refer to it as a distinct chromosome. It's now visible. You can be able to say this is a chromosome. And number two, the centrioles have now formed the spindle fibers, begin to form the spindle fibers. But most importantly here, at this point in time, there is the earlier prophase and the late prophase. As the process continues to move to the late prophase, now the nucleolus begin to disappear. Number two, the nuclear membrane also begin to disappear. So that is very important to note. After prophase, we go to metaphase. Now, at metaphase, you cannot be able to... I've already told you at prophase that the nuclear membrane begins to disappear and the nucleolus also begin to disappear. So at metaphase specifically, there is no nuclear membrane, there is no nucleolus, and therefore what you are now able to see here is just chromosomes aligned at the center. We refer to them as equator alignment. So they are aligned at the equator. So you can tell that this is a metaphase by looking at there is no nucleolus, there is no nuclear membrane, but that is not enough. The most conspicuous reason is that the chromosomes are aligned at the center. So in an examination, I'll simply give you a question or a diagram like this one, and I'll now ask you to tell me the stage of mitosis this is. And therefore your answer should be, which is metaphase. Reason should be, chromosomes are aligned at the equator. After this point, remember we are dealing with chromosomes. At the end of this stage, we need two daughter cells. So now proceed to anaphase. And at the point of anaphase now, you can be able to see here. As you can remember, I had given you a structure of the chromosome, which is here. And it has two chromatids. But these chromatids can separate at the center here. And if they separate at the centromere, they now give us how many chromosomes? 2 chromosomes. Those 2 chromosomes are now achieved at this point. They are achieved at this point because there is, of course, a role of activities which are happening in this point, which is involving shortening of the spindle fibers. Now, at this point where the spindle fibers are, after they have broken or separated the chromatids into 2, the chromatids will migrate to the opposite poles. One move to the North Pole, another one to the South Pole. Of course, they can be South, East, and West. So we don't encourage the use of North and South, East, or West. We say opposite poles. Now, after this has happened, in an examination again, you can be given a diagram like this. Which stage of mitosis is this? The answer should be anaphase. How did you know? How you know that that is the answer is you will tell us that the chromosomes are broken and separated and chromatids are migrating to the opposite poles, marking point. And then after this has happened, now the end stage, which is telophase. Now here, the outermost part, which is the cell membrane, you can see it is now beginning to shrink in the normal language. It's beginning to come inside. But before you understand that, I told you earlier that there are 2 things which happens before you break something into 2, which is a cell. A cell with the nucleus starts with the division of the nucleus and then the division of the cytoplasm. So here, inside first, the nucleus will divide. And after the nucleus divides, we call that as karyogamy. It is followed by cytogamy, which is the division of the cytoplasm. So here, at telophase point, the cell now begins to constrict. We call that as constriction of the cell membrane in an animal cell. And therefore, once it begins to doing that, finally, it will break and give us 2 daughter cells. Don't ask me why they're not giving you 2 sons. But you must be very keen that at telophase, there is the return of 2 things. One, the nuclear membrane has returned and it's reappearing. And now, what happens here at this point is that also the nucleolus has reappeared. That means this cell now cannot be able to be seen. The chromosomes are not visible. They are even appearing like interphase. So after telophase, the cell enters again into another stage, begins afresh into interphase, after forming 2 daughter cells. This cell constriction is happening in animals. If this was a plant cell, then a plate like this one I'm drawing here will appear, and we call that one as a cell plate. Formation of a cell plate in plants for an animal is constriction by the cell membrane. And that marks the end of our lesson. But before we end our lesson today, I want you to try this assignment and try to give us what do you think is the significance of mitosis. So you will try this question, explain the significance of mitosis in living organisms. Of course, this mitosis, of course, is happening in real life situation. You know, many students ask me, does this happen? It's happening in real life situation, in a yeast that is budding, in amoeba which stays in water, that is by innervation. In vegetative propagation, you have mitosis happening. And now that I've already told that on the Malpighian layer, which you already have as a human being, undergoes mitosis, it is really happening. If, for example, you are bitten, someone maybe cracks your skin, and then after some time, it has to heal. Those formation of new cells, they have to do so by cell division. So that's real in life situations. It is happening, and we are dealing with a situation that is real in real life. So this is how mitosis occur. And next episode, we'll be looking at meiosis, God willing. But before we look at the next episode, please remember to subscribe to our channel. Our channel is Evans Matuga. Please click subscribe. See you in the next lesson.